All right, so in this video, I'm going to be going over specifically just the carbohydrate component. Um, so our focus really is going to be about what do we really need to focus on carbohydrates. This is going through the different names of if it's a monosaccharide versus a polysaccharide and kind of what the purpose is for each one. But first and foremost, you want to know the biggest component about carbohydrates and what you're going to be probably tested on is their use. And that's really going to be used to be broken down in cell respiration to make ATP, or they're going to be synthesized in photosynthesis. So the first part about this video is really going to be about what is a monosaccharide. And the most one that we're going to be more familiarized with is going to be glucose, which is by far the most common, but there's other weird ones that we have to know is in terms of galactose and fructose. You're not going to have to know the names of those, but it's typically just going to be a single carbohydrate. And when you think about the function of it, you're familiarized with kind of the structure of it looking very nice and cyclical like this. But again, as it's stating here, it's the major fuel for cells. And when a plant is building glucose, this is typically what they're making. They're making a single monosaccharide and then it gets linked together. So when we hear that word a monosaccharide, mono meaning one, it's just one of these carbohydrate molecules. However, most things are going to be multiple carbohydrates linked together, and they're held together by what is called a glycosidic linkage or glycosidic bond. And one question is that is often throwing students off as well. If this is one complete carbohydrate, C6H12O6, how come if we have a disaccharide, why is it not just doubled? Well, and the biggest thing is that why isn't it C12H24O12? Well, remember, anytime you make a bond, you actually lose water. So this is just showing that same video where if you lose this hydroxide, if this is C6H12O6, this is going to be also C6H12O6. And when you look at this, you've lost water. That's why your hydrogen and your oxygen goes down. And this right here is your glycosidic bond. So similar overview. You're not going to have to know the specifics of the names of like a disaccharide. But if you lose water, again, it's following that logic. It's a dehydration reaction. You form your bond here. So in terms of that, if you have a disaccharide, di meaning two. If you have multiple carbohydrates linked together, it's used for really long-term storage. That's what we're going to call a polysaccharide. So in terms of that, this is showing you a nice overview. Many glycosidic bonds form together. So this is showing the difference between a couple of different polysaccharides. You have starch, glycogen, and cellulose. Starch is going to be a form of storage for plants. If you think of like the roots of a plant, this is when they would typically need to hold all this starch in because that's where they're doing cell respiration. We store starch as glycogen. They look almost identical in terms of their glycosidic bonds are going to be very slightly like if you look at cellulose, notice that there's this bond that's facing upwards and this glycosidic bond is facing downwards. So this is just kind of the overview. You're not going to have to know the difference between these two, amylose versus amylopectin, but you do need to know plants can store their carbohydrates as starch. Typically, they're going to be found in the roots of those for sure. So when you think of where this gets stored, it's going to be in the roots. So, sorry, it's just on the trackpad. So starch is stored in the roots. We store all of our carbohydrates as glycogen. That's where we keep it all nice and stored. So typically, if you have a big diet full of carbohydrates, you're going to store that in the liver until needed. So this is just showing you a different overview between the starch in a plant cell versus the glycogen in your liver. So slight difference between those glycosidic bonds. And you might not think that there's a difference for you guys in terms of these polysaccharides, but this is just showing you the difference between an alpha and a beta glucose. Alpha has a hydroxide facing downwards, glucose facing upwards. The biggest thing about this is you actually can digest starch because of the way that that glycosidic bond is here. I have another glycosidic bond here, another one here. If you look at the beta glucose, it forms these alternative glycosidic bonds and you actually cannot digest it. So all of the cellulose that you would eat in things like celery or lettuce, for example, you don't really get any caloric value from it because it just passes through as fiber. So it's one of those weird, interesting things that the shape of your enzymes, you can't actually break them down. So in terms of that, this is just kind of summarizing what I was just mentioning, is that we biologically can't break it down. We do have some microbes that can help digest it, which are the bacteria that you would have, but just something interesting that we biologically don't make the enzymes that can break it down. Chitin is one of those last interesting carbohydrates. It's typically going to be used for the exoskeleton. It's really, really firm. It doesn't fall underneath. It has more than carbon, hydrogen, and oxygen in its structure. It does have nitrogen, but this is not something that I see commonly tested, but it is made up of like the hard exoskeleton of a lot of arthropods. And that concludes your short recap on carbohydrates.